Okay, so I'm going to tell you quickly about what has happened over the last 3 years here in my part of the operation in Singapore. I'm going to be talking about complexation and bioavailability and bioavailable concentrations of zinc and cadmium in Singapore coastal waters, the Malacca Straits, and the southern South China Sea. There's a long list of collaborators. Most of this work was done with the help of Yanni, Meng Li, Asani, Moritz, and a bunch of other people as well. So there's a lot of people to thank. Importantly, Ed just gave a talk, and you guys should go and take a look at the posters that Meng Li, Moritz, and Xiu Qin are setting up about work in mangroves. A large riverine project we have in Malaysia, and some of the lead story as well. Those are the collaborators, the laborators now over the last few months while I was in the hospital, were my very faithful students. Caroline, Jiawei, and Jui were doing quite a lot of work basically on their own while I was recovering. Okay, now let me go to a brief intro, which is most of you must have heard already a few times. Well, I'll try to go quick. We study trace metals because they're important for for enzymatic requirement to the cell. Here we have a cartoonized version of carbon, phosphorus, and nitrogen uptake, and they are mediated by enzymes that use these metals. Iron and zinc notice that zinc is used for the carbon and the phosphorus uptake. Cadmium is here and can replace, as well as cobalt, zinc. So we care about the bioavailability because of the total concentration, of the total dissolved concentration of metals. We need to know what are the fraction, what is the fraction that is actually going to make the biological effect. So we know that the pool of total dissolved metal can be broken between inorganic, free, and organic compounds, but in nature, largely this is dominated by these ligands, which basically means that the total dissolve is mostly as an organic complexes, which is mediated by the really strong binding strength with which this chelating agents chelate to the metals. The problem being that these two are the portions that are bioavailable. So if you're talking about a metal that has a nutritious role, you could be starving of something for biological purposes, even though you have a large concentration. Now, if it's a toxic element, it can be a good thing, right? Because most of it will not be bioavailable, and a small fraction only will be bioavailable. So we've been studying this under the common assumption, sort of the paradigm of these ligands being produced in surface waters and the vacuum, sort of in the open ocean. And my stay here in Singapore was basically to try to prove that was not the right assumption, that we should actually take a look at not only industrial sources of metals, but riverine matter as being important sources of humics. So basically, in my master's and my PhD, I did a lot of work in the Pacific and the Atlantic, and lately in my postdoc in MIT, I did work in the Middle East, and we basically found that the binding strength of the strongest of the ligands that binds to zinc, around 11 and a fraction, these are in log scales, basically for the Atlantic and the Pacific is what I found in some of these places that relate to water masses that come from distal sources of riverine material. And in my time in MIT, with Ed, we went to Kuwait and we did some work in the Tigris and the Euphrates River directly, and we found exactly the same strength of the binding agent, proving that the rivers, the riverine end member compares really well with distal riverine signatures. So the idea was then to try to see that in this region, which has so many rivers and so much organic matter coming from these. So over the last few years, we've been basically working pretty hard at setting up a trace metal clean lab at SMART. We did that with some funding we got from Sensam in 2015. And then in 2016, we sampled all over Singapore and a little bit of Malaysia, sampling for seawater, river water, and corals. And then we also did quite a lot of work with Janelle trying to set up sites for harmful algal bloom sites around Singapore. In 2017, we have 3 projects that we are still working on. One of them related to the different monsoonal time of ligands and metals around the region at a different point in time. And then we also have one mangrove project that we sort of piloted last year. And then we are working on this coral project that I'll tell you a little bit more later, with the idea of basically being able to understand the ligands now and in the past through coral records. So we can actually look at the past and look into the future. Basically model ligand and metal bioavailability, how they control the metals, and then how that will change in future climate scenarios. The main questions then are, at the regional scale, we want to know if the ligands of humic origin come from rivers into the sea. How do these ligands change interseasonally? At a local and zonal scale, so for Singaporean interests, we want to know how do these local sources of ligands, which can be related to phytoplankton or seawater in general, traversing through the Singapore Straits, harmful algal blooms that can be found in the Johor Straits, or mangrove as sources of organic matter, how do these respond to metal sources, which are pretty diverse in the Singapore region, in the Singapore local scene. And at both scales, we want to figure out what is the primary regional regulator of bioavailability, how large is the regional trend versus the local influence of diverse factors, and what can we infer from humics recorded in corals about ligands through time, so that we can understand about that. And we can actually get an assessment about riverine organic matter controlling metal bioavailability in the past and in the present and in the future. So basically, we're trying to look at the same question from 4 different scales, but it's basically the same question. I mean, whether it is a regional study, a local study, a zonal study near a few meters up or down a mangrove, or a microscopic study, we're basically looking at the same question. So we chose Singapore because Singapore has a diversity of places to study all sorts of things. You can actually find very polluted, from a trace metal perspective, right? I mean, shipyards and oil rig platforms in the urban areas, and also pretty pristine places, and also a lot of fish farms, a lot of industry towards the west. In the Malacca Straits, we did work in separate trips, looking at the Malacca Straits and the South China Sea. And we basically got this high-resolution sampling scheme done last year, which basically finished the day before I broke my leg. The Malacca Straits is a really interesting place in the South China Sea, because we have everything between pretty riverine influenced environments, relatively clean places, but also a whole lot of agricultural activities related to palm oil. So basically, I'm going to be talking about these 4 types of the directions of results: metal concentrations, bioavailability or non-bioavailability of the bioavailability and non-bioavailability distribution of metals, the seasonal changes, and the potential sources of trace metal contaminants from a biological perspective. First, to start to talk about trace metal sources, I will tell you briefly that there are natural sources that change through time. Here we have at Kusu Island some oscillation in terms of other elements, and in other stations around Singapore, you can see that there are some anthropogenic sources that are much larger than the natural sources. Ed mentioned a couple of things about lead sources, and if you want to know more about this, you should go and take a look at Meng Li's poster. In terms of the ligands, which is sort of the meat of the story of my presentation here. Now, basically the idea is this: on all of these places, we initially chose these 3 sites to take a look at the binding strength of the chelating agent, to see if they do compare with the ligands that we were finding remotely. And we did find that the ligands here in the Malacca Straits at Port Dickson were basically in the same order of magnitude of binding strength. These are here in the narrowest section of the Malacca Straits. So the water basically narrows here all the way to the narrowest point, and then it opens up into the South China Sea in something that I refer to as a Malacca sort of funneling effect. Now, looking further at more samples, we did have some samples that I sort of cleaned up the data a little bit to try to figure out and get some trend. From all of these samples, trying to filter out samples that would be too influenced by other factors. So we got to a point where it would be more or less linear going up and down, getting into the highest point around the Malacca Straits and Port Dickson. Now, what we did over these last few months, well, what my students did was run a lot of the analysis for all of these other sites. Here we have Port Dickson here. They did a lot of the Malacca Straits work, and then they did a couple of extras on the South China Sea samples. And then they did also a whole lot of, this was the challenging part, doing a lot of these Rajang samples from around here, from one of the biggest rivers in Borneo. So what we found was basically that the same trend prevails with a little bit of noise, yes, but it basically goes up and then it goes down. And that, but more importantly, rivers are much larger source of ligands. So here in the Malacca Straits, there's a couple of small rivers, but then the larger rivers are the Pahang here and the Rajang and the Igan here. So we need to study some of those closer. If you want to hear about that, you should go take a look at Moritz's poster talking about the Rajang project. So again, if we filter out a little bit the data, we basically get a similar trend going up and down going up on the Malacca Straits and going down on the South China Sea. Same funneling effect. If you look at cadmium is a little bit more messy. I mean, we got some data initially, but then we started to get the high resolution data. It is a little messy. I mean, there's a lot of ups and downs. So we were confused. The river is not clear either. So in fact, we basically end up saying that the ranges are relatively large in the Malacca Straits section. The range is relatively smaller on the South China Sea section, and it's pretty variable, meaning that there are some ligands and there's some excess metal on the Rajang River. So we basically need to study much more the river systems. We found some seasonal changes as well. We studied this. We haven't done it on the Malacca Straits. We've done the seasonal changes only around Singapore. And what we found was that on these 5 sites, and I will go over each one of the 5 sites pretty quickly, the range is huge. We go from 0 to 400 nanomolar zinc and all the way to 1 nanomolar cadmium. And there are some that are excess metals, some that are excess ligands. So it's a really confusing, apparently, place. But if we go one by one and separate each one of the stations, here we are at Kusu. Kusu sees only ligands for zinc and only excess cadmium, meaning that probably this could be sort of the natural scenario whereby cadmium is upwelled and changing through time through the monsoonal seasons, and the zinc ligands are changing also within a relatively narrow range between 3 and 5 nanomolar. We are actually continuing to try to do this, looking at organic matter coming from Sumatra peat in a project that we're working with Patrick Martin from NTU. If we look at other places, we found actually almost by accident, it was not the point to do this, but we found evidence of logarithmic trend, decreasing concentrations of zinc and cadmium, which we think to us are related to land reclamation settling down. In a couple of other places in the West Coast, we found, West Coast was an interesting place because West Coast is right here, right next to a big shipyard operation. And we found that there was excess of metals for both zinc and cadmium in 3 points in time, but at one point in time, there was a lot of ligand for cadmium and also a lot of ligands for zinc, implying that this was a point where the environment was sort of responding to the environmental injury that this load of metals was meant. We actually do have a mangrove station report from 100 meters away from here that is discussed in Suchin's poster further down. If we go to a couple of other places, Kranji and Pungol were interesting places where We talk about fish feed, so basically fish farm operation, meaning a source of metals that I really wasn't counting on. I thought that this would be a source of organic matter, but it was a source of metals instead. So the question of bioavailability and bioaccumulation comes up. We're probably going to be working on that later on. The question about corals also, I'll try to be super brief about this. Ed introduced the idea of the coral bands. Here we can see them bioluminesce with UV light, and we can actually quantify the amount of humics that are accumulated in each year. And with this idea of humics, if we could quantify, we could get an idea about ligands, as the ligands and humic matter sort of relate. And more so, since Yanni has, this is from her 2017 paper, where she has a beautiful study of all the rivers around the Peninsula Malaysia. The seasonality from river data and trying to compare it with what you find in luminescence in the corals. So in a nutshell, what we wanted to do was we wanted to study in this region the influence of rivers on the marine environment. And yes, we have done that for the last few years. We are actually quite happy to see that rivers are important, and we need to figure out what biological agent or where do the ligands come from if they come from rivers. If there's any ligand being produced or brought into the system, what ligand is it, if it's a strong ligand or not, and then how is that going to affect the zinc bioavailability and the distribution of ligand of zinc between different pools. So in conclusion, I'll read this briefly and then we can finish up. This was a study of trace metal concentrations and bioavailability regionally and locally. We looked at zinc and cadmium sources, and we found that there's natural sources of metals that are much smaller than anthropogenic sources. Anthropogenic sources included shipyards, land reclamation, and fish farms. In terms of the ligands, the ligands for zinc is locally connected to regional and global distribution of ligands, so I'm pretty happy about that. There is this funneling effect along the Malacca Straits to Singapore to the south end of the South China Sea. There is a really large riverine influence, which we are really intended on continuing studying in the Rajang mostly, and in a couple of other places in Peninsular and Malaysia and Borneo as well. Cadmium is not too clear, so we really need to do a little bit more work on this. In terms of the time variability, the natural oscillations along the Malacca Straits, Singapore, South China Sea can be related to the physical transport of the ligands along this transect. We're going to be a second time point now in the second part of 2017. We'll get at different time points we can see about the monsoonal effect. But we're very excited about this coral project where we can look at longer time scales. And that's all I have to say. Thank you very much. It's been a fun ride.